Hello, my name is Maria Stephenson, and I'm a co-op student at the Dr. Nash Bureau Lab at Canada's Michael Smith Genome Sciences Centre. Today I will be presenting Rna Scoop, a tool which provides an interactive visualization of isoforms in single-cell transcriptomes. Single-cell sequencing has become a powerful tool, allowing researchers to examine tissue heterogeneity and characterize rare cell populations. However, the majority of single-cell transcriptome analysis pipelines focus on the quantification of gene expression, ignoring information at the isoform level. Isoforms are RNA transcripts from the same gene to different sequence. These differences could be a result of RNA processing events, such as alternative donor and acceptor sites, exon skipping, or intron retention. They could also result from alternative transcription start and termination sites. Because isoforms differ in sequence, their functionality can differ as well. And it is not rare for a gene to produce multiple isoforms. In fact, 95% of human multi-exon genes are alternatively spliced. Because of this, isoform detection and discovery in single cells is important, and could advance our knowledge of tissue heterogeneity and cell diversity. We created Rna Scoop to allow researchers to easily visualize isoforms in single-cell transcriptomes. With it, users can view the structure of isoforms, identify their expression levels in different cells, and compare isoform expression across clusters. To achieve this, Rna Scoop consists of two major components, an isoform view and an interactive t-SNE plot. Now, for those of you who don't know, t-SNE is a technique to reduce high-dimensional data into lower-dimension representations. In particular, a 2D representation can be plotted easily and has been used widely for visualizing single-cell RNA-seq data. I will now go over the basic features of Rna Scoop. The program is written in Java, and its GUI is implemented with JavaFX. The program consists of the isoform view on the left, t-SNE plot on the right, and activity log console on the bottom. The input to Rna Scoop is a single JSON file, which specifies paths to a GTF file containing the isoforms of interest, a matrix file containing their expression levels in each cell, and files containing labels for the matrix rows and columns. The dataset I'm displaying right now is a collection of 3,840 cells from the Tabula Muris compendium. Users can select genes for the isoform view, where all isoforms of selected genes are displayed. It's important to note that the genes can be sorted based on maximum fold change across clusters in the t-SNE plot. This allows users to easily identify differentially expressed genes. In the isoform view, isoform representation is similar to that used in UCSC Genome Browser and IGV. Exons are represented by boxes, and lines represent spliced introns. By default, isoforms are drawn according to their exon's chromosomal coordinates, as if the corresponding genes were on the positive strand of the genome. However, users can choose to reverse complement isoforms on the negative strand, so that they're drawn in their 5' to 3' orientation. This can be useful in cases where gene orientation is known. Additionally, in order to hide potentially incomplete isoforms and noise from sequencing, we give users the option to hide single exon isoforms. The isoforms are colored according to their average expression in the cells in the t-SNE plot. Viewing the gradient by which these isoforms are colored, we can see that the H3F3AOS isoform has an average expression of around 100 in the cells, and if we hover our mouse over the isoform, it will tell us what its average expression is. Now, it's important to note that the gradient is completely customizable, allowing users to change the TPM cutoffs, change the colors of the gradient, and change the scale of the gradient from linear to logarithmic, and vice versa. The t-SNE plot is completely interactive, allowing users to zoom in and out of different areas and pan the plot freely. Cells are selectable via lasso selection. When the user selects a group of cells, the isoforms are colored according to their average expression in the selected cells only. So for example, if I select this cluster of bladder cells right here, I can see that the H3F3AOS isoform is expressed in them, but the TMEM119 isoforms are not. In addition, isoforms are selectable as well. When a user selects a group of isoforms, the cells in which those isoforms are expressed are selected. So for example, if I select these TMEM119 isoforms, I can see that they are mostly expressed in the microglia cells. This makes sense because the TMEM119 gene is known to be highly and exclusively expressed in microglia. With these features, users are easily able to view isoform structure and identify isoform expression levels in cells. However, analyzing expression between clusters is still a bit awkward. For example, let's say I wanted to compare expression of these isoforms across all cell types. Currently, I would have to select each cluster individually, and that could get really annoying. For this reason, we introduced an important feature, the dot plot. The isoform view has an option to show a dot plot next to each isoform. Each dot is colored according to its isoform's average level of expression in its cluster, and its size represents the percentage of cells in its cluster which express the isoform. So now we can clearly see that the H3F3AOS isoform is expressed across all cell types, and we can see again, perhaps more clearly this time, that the TMEM119 isoforms are only expressed in the microglia cells. We can also see how the expression of these isoforms varies in the microglia cells, with the first isoform having the lowest average expression, and the second isoform being expressed in the lowest percentage of cells. Now when cells are selected in the t-SNE plot, the values in the dot plot are calculated based on the selected cells only. So for example, if I select this cluster of microglia cells here, I can see that the dot plot has changed. The last feature I want to talk about is the ability to switch between multiple label sets for the t-SNE plot. Users can create their own label sets via cell selection. This is done by clicking on the Change Cluster Labels button and selecting Add New. Selected cells can then be given custom labels and colors. New label sets can be saved, and users can switch between label sets at any time. This option is important for two reasons. The first, it allows users to view the t-SNE plot at different levels of detail. The second reason is that it allows users to easily compare expression in any group of cells. For example, now we can clearly see how the expression of these isoforms varies in the different microglia clusters. So in summary, RNA Scoop allows researchers to visualize isoforms in single-cell transcriptomes. With it, users can view isoform structure, identify isoform expression levels within cells, and compare isoform expression across clusters. Lastly, I would like to thank all my colleagues from the Dr. Rna Bureau lab for all the hard work that they've put into this project, especially Calming Nip, who has contributed equally in this work. This project has been funded by Genome Canada, Genome British Columbia, and the National Institutes of Health, and my funding for this conference was provided by ISCB through their Fellowship Award. Lastly, I would like to thank you for listening to my talk. I have a list of posters from the lab if you want to check them out, and I'm happy to take any questions.